2.6, you guys, is talking about Mendel's second law. His first law, does anybody remember the name? Law of, it starts with an S. Segregation, very nice. His second law, and only law, he has two laws, is the law of independent assortment. Okay? So that's what this section's going to go into more details with. And what it is the alleles separate from each other into the gametes, and they're independent of each other. So it's just random. And that's due to just the two chromosomes coming apart, right? Or the two homologous pairs coming apart. And then also crossing over. So let's get down fertilization and meiosis. Okay, so fertilization is just the sperm and the egg coming together, fusing into a zygote. Now, that's if they have sperm. Land plants don't have sperm. What do they have instead? Pollen. So fertilization would occur down here in the ovary. I talked to you guys about this flower last time, right? Between the pollen and going down in the egg is waiting. Okay? What are the egg and sperm called? What type of cells are those? Or the egg and the pollen? Either one? Gametes. Or more commonly, sex cells. You guys remember this? Okay, now, so it's just a random chance, you guys. Which pollen got blown on to the stigma and successfully went down the tube and fertilized an egg, right? First come, first serve. Even though it's been pretty interesting, there's been a lot of research going on with sperm competition. They have actually seen where sperm are fighting, like a race to get to the egg, and they even see them drafting. Do you guys know what drafting is in racing? You can do it, some of you runners or maybe have done it, but you see it a lot with the race cars and stuff. So they will see, sperm will it can't be intentionally, they don't have brains or whatnot, but they will be stuck behind a group of other sperm dragging them with them, okay? And then on a turn, a rebound or something, I don't know, they shoot around it. Some of you might know this better than me watching racing. So there is already survival of the fittest going on. I mean, if you got a sperm that has a mutation, right, and it's going in circles, that one's not going to pass on its genes, right? So it is random. However, there is some sperm are in better shape than others. So already sexual selection is happening here. Natural selection. Pretty cool. Okay, then, so when I ask you guys, what makes us so different? What makes the diversity? One is the randomness of just fertilization. The next one, it happens with meiosis and just the chromosomes separating. And then there's a third thing during meiosis that makes things all mixed up and randomized. What is it? What's it say? Crossing over. Yep, you're right. It's not here. I'm pointing at something invisible, aren't I? Crossing over. So that's our next term, correct? So first, 50-50 odds if you're going to pass on mom's chromosomes or dad's chromosomes, and then crossing over. And after you get the bulge, you're going to want to sketch. So remember when we did the meiosis square dancing, the model, and I told you guys that also the homologous chromosomes, when they get next to each other, when they synapse, it's called, they can exchange pieces of themselves. Do you guys remember me talking about this? That's called crossing over. So that you're a combination of your parents' genes that came from your grandparents. And you guys, have done meiosis in your gonads. So your mom and dad's chromosomes have crossed over inside your cells and then separated, waiting in those eggs or sperm that you guys have made. And that's why siblings can be so different from each other, because of this randomness of what allele do they inherit. So this is called what again? What's that called, you guys? Crossing over. Good. Okay, crossing over. And crossing over increases diversity in offspring. Can I go? Okay, so let's get that down and I'll explain the bold. Here we go. So, this is kind of abstract, but it's pretty cool. You guys with me? Genetic linkage just means that genes can be linked. This is in contrast. This is not what Mendel was saying. Mendel was saying that genes are inherited independently from each other. Well, here's some evidence that not all genes are like this. And it makes sense. It depends where they're at on the chromosome. So look up here. When crossing over occurs, this thing can break in all these spots, all the way down. Do you see all the places it can break to do crossing over? What are the odds it's going to break just right between these two genes here? I can't get it. You guys follow? Look, I can't even get my pencil between the two genes. You guys following me? So odds are genes that are close together on their, what is this, chromosome, have more odds of getting passed on together. Does that make sense, you guys? Hello? You guys with me out there? Now, according to Mendel's law, between any pair of these genes, like D and B, are there a lot of chances they're going to get separated from each other? B and C. All these places, it could break. And it does. And that's why we see independent assortment. I'm not sure if Mendel's 7 traits, genes he was following, are all on different chromosomes of the pea plant or not. If some of those are on pea plant, you guys can do this for bonus if you want. Find out. Okay, where are the genes, the 7 traits Mendel followed on his pea plants? What chromosomes are they on? Are any of them on the same chromosomes? This has been well documented, so I guarantee that we have sequenced the genome to peas also. So we know, just like with our human chromosomes, we know where all our genes are. Okay? So if you want, find that out. Don't forget your resource and find out. If they were on the same chromosome, they must have been far apart from each other because all of his data shows that they were split up all the time. Does that make some sense? The other thing I want you guys to realize, you drew that little picture, that when the chromosomes and the homologous pairs do come up next to each other, there can be several crossing over events. So those can break on the top, they can break on the bottom, then even after they've broke again, they can break again. So once they're next to each other, the DNA is very fragile, which is a good thing because it allows it to be broken up and split apart and all that easily. So kind of interesting. So again, if they're close together, then a lot of times they're inherited together. You guys listen to me here. If they're close together, many times they're inherited together. Okay, and if they're inherited together a lot, they're called linked. Does that make some sense? Okay, good. All right. So, we're not done with chapter 6, but I'm moving on to some other stuff here in chapter 7. We'll go back. But chapter 6 is all about Mendel's patterns that he discovered. Chapter 7 is there's other patterns. The little goofy video on YouTube talked about that, right? A lot of them are not, a lot of genes don't follow Mendel's laws. There are a bunch of different factors that affect what people look like or the traits they get. Remember that word phenotype? Okay, so you guys already have incomplete dominance down. So incomplete dominance is totally different than what Mendel learned and shared with the world. Incomplete dominance makes more sense. This is what other people were saying, was blending. Do you guys remember? You are blended from your mom and dad. And for some traits, that's true. Here's the Siamese fighting fish. If the heterozygous is a blending of it. So there's a blue allele and there's this greenish allele. And if you get the hetero, if you got an allele for blue and an allele from the green, You're like an intermediate, an in-between. Another famous example of this is with pink carnations. So what do you think the parents of pink carnations are? Homozygous red and homozygous white. And now the hetero is obviously a blending of red and white mixed together as what? Pink, okay? So there was some examples, and there still are some examples of the blending in some genes. Okay, what's it called, you guys? Hello, what's it called? Incomplete dominance. There's one of your vocab terms. And there's an example. Now, codominance is when both alleles are being expressed equally. Okay? So, for example, here is blood type. And you have an A allele possibility, a B allele possibility, or no, neither one of the A or B. Okay? Now, what is the A and B? The A and B in your blood stand for a protein. I have A blood, so I know I have one A allele, and that means I have what we named the A protein in my blood. Does anybody know if they're B or AB in here? Well, if you're B, you have a little bit different protein, and we named it the B protein in blood. The O actually, I think, should be a 0, because what does it mean? They don't have the A or the B protein. So this is called codominance because they're both being expressed. What does co mean? Are Hager and I co-teachers? So we're both contributing, right? That's the idea here. So now this gets a little more interesting with blood type, okay? I said I was what type? A. Do we know my genotype? We know for sure one of them. We know what I don't have, but I could have an AA or what? An A and a, oh, okay, cool. Sometimes they do call it the I. So that's used also. It just means it's missing. It's the same thing as I could have wrote this. I don't know what this is. Okay, but I do know though it's not B, right? So we don't know if I'm this or this. Okay? Now, what about if you're B? Same thing. You're this or BO again. Okay? No, you don't stink. Okay? Do we know for an AB? An A and a B. There's their genotype. Okay? And then O is neither. So they have to have this, right? Now, when this gets interesting is for if you guys give blood or, you know, work down there at the blood drive, or you go with friends or your parents or whatnot, ask them what blood type you are. It's not as important as it used to be, but if you're hurt, if somebody's hurt and they need blood, if it's a real emergency, what type of blood are they going to give you? They're going to give you this one because they don't know what you are. Okay, so people that have this blood are in high demand. The blood bank calls them and asks them regularly to donate. Okay, now there's something I'm forgetting. Does anybody know what I'm not mentioning? What else about blood type? O does happen to be the most common, yeah. Right, so can O take from anybody else? No. So they're the universal donor. Who's the universal recipient? Can I take everybody's blood? I can take this blood, or I can take this blood or this blood, right? What happens if I take this blood or this blood? Yeah, I have an immune response because all of a sudden there's all these blood cells in my body, the B ones, that my body's like, what is that? And starts attacking what's supposed to be trying to help me. Okay, so that's why we can't do that. But I'm still forgetting something. Nobody's heard about a blood type that I seem to be forgetting something about? The negative, yeah. All that is, that's called the RH factor. It's just another protein that people have in their blood. Diversity again. If they're positive, they have it. If they're negative, they don't. And I actually am a positive. Okay? So, anyway, just kind of fun and interesting. If people have health concerns, they often carry their blood type on their wrist. But nowadays, like people, we were told when I was a little kid, you should know your blood type because if you get sick, you can tell them. You know, and that's why you use a bracelet in case you're hurt and you can't even tell them. But nowadays, they won't believe you what you tell them. They're not. It's not worth the chance unless they're low on this. But in a real emergency, they're just going to give everybody this. And then they'll type it, and then they'll switch you to save, you know, the blood bank and give you only what you need, you know, what you can take. So kind of cool. What's that called? Codominance. And in this case, it's also known as, because you could have the A allele and the B allele or the O allele. And then coming together, we get how many blood types? 1, 2, 3, 4, and if we mix the negative positives in there, we get even more. Okay? All right, another one is called polygenic. So the other one is you got more than 1 or 2 phenotypes. Here you definitely get more than 2 phenotypes, but what's causing it? How many what? More than 1 gene. Okay, that was not the case with the A and Bs. Okay, so now we have the phenotypes are combinations of many genes. What's the name of this vocab word? Polygenic. Poly means many, genetics, genes. And your eye color. And when we do baby face, I love that. We'll do that next week in the beginning. You guys will make babies, little baby faces, and you'll get to, you know, look at this and see all these genes. Oh, skin color's another one. Okay, epistasis. And some of you maybe had trouble finding epistasis. So epistasis is when another gene interferes with another gene or affects another gene. And you'll have one of those on your baby face also. So one gene allows another gene to happen or not happen. Okay, some genes will turn other genes off. Some genes will turn other genes on. So the genes here are working together, affecting each other. That's called epistasis. Okay, we good? Okay, the last thing is the environment. And for me, I think this is kind of the most interesting. One of my favorite examples is the flamingos. What color are flamingos? Pink, and mostly actually white, and it depends. The ones that are really pink are eating a lot of what? Does anybody know the story? It's the krill shrimp. You know, when you guys see shrimp, are they kind of pinkish, orangish, pinkish? And when you get next to a flamingo, they're orangish, pinkish too. But it depends on the shrimp. If they get that molecule, those ingredients from the exoskeleton of the shrimp, it accumulates in their feathers. So the environment is giving them a phenotype, a physical characteristic. And you guys know same thing with skin color too. If you spend a lot of time in the sun, what happens to your skin? It gets darker. You make more melanin. You make more pigment to help yourself. Okay? Now this one is pretty interesting. Turtles, and there's a few reptiles or amphibians that can do this. When the mama lays the eggs in the beach and buries them, if the temperature is different, I think it's if it's hotter, they're boys. The babies will become boys. If it's colder, I think, does the book say this? Did you guys read this in your, I can't remember the example. But anyway, the temperature affects what gender develops in the egg. And it has to do with the chromosomes. Like bees, you guys have heard this? To make a queen bee, you have to feed these babies special, what does she call it, royal jelly. They make special food for the babies, and then they develop, it affects their genes so that they develop differently than the other ones. All ants and bees, or most of them, are boys. Did you guys know that? Then if we need females in the population, it's like the chromosome doubles, and it's like the two X's again, make a girl. But the boys are just one X. They don't even have a Y. They just have the one. They're haploid. They have one of each. So the environment can affect what the organism's gender is and also its looks. Hydrangeas flowers, you guys might know that. That has to do with how much acid's in the soil. If they're, I think it's reddish or if they're bluish. So pretty interesting. Height. You guys realize that humans have been getting, on average, taller over the last few hundred years. You can see evidence of this. You go up to Virginia City or historic places. The doorways are smaller, the beds are smaller, people are smaller. Okay, you guys survived.